Hello, welcome to the July edition of the Nutritionist webinar. I am Mary Ann Fessenden from Agricultural Modeling and Training Systems and serve as your American host. The purpose of these webinars is to provide access to technical seminars on a range of topics delivered by internationally recognized speakers. The series is a unique three-language presentation held in English, Portuguese, and Spanish. Noted ruminant nutritionists will present in English, hosted by AMTS in the United States, while Marcelo Hens Ramos from 3R Lab simultaneously translates into Portuguese for Brazil and Paula Torillo translates into Spanish for Argentina. Ordinarily, there is a post-presentation question and answer period during which listeners can submit questions through me or Marcella and Paula. Unfortunately, during this month's seminar, we lost that recording, so there will be no appended question and answer period. We will instead proceed with the webinar. Professor Randy Shaver was born and raised on a western Pennsylvania dairy farm. After completing a BS degree in dairy science at the Pennsylvania State University, Randy received an MS degree from the Department of Animal Science at the University of Maryland. He then completed his PhD degree in dairy science at the University of Wisconsin-Madison in 1986 after conducting research at UW-Madison and the U.S. Dairy Forage Research Center. Randy spent 1986 through 88 working in the industry. Since then, he has been on the faculty of the Department of Dairy Science, University of Wisconsin-Madison, with a 75% extension and 25% research appointment. Randy's extension and research programs focus on applied nutrition of lactating dairy cattle. He has advised or co-advised 29 master's or doctoral students at UW-Madison. Randy has authored or co-authored 88 peer-reviewed journal publications, 170 scientific abstracts, 101 popular press articles in industry trade magazines, 249 newsletter articles, extension handouts or bulletins, and internet publications. He has presented 590 invited papers at industry conferences in 46 states. Additionally, he has been an invited speaker for dairy producer and feed industry audience in Argentina, Brazil, Canada, 9 provinces, Chile, Colombia, Czech Republic, Denmark, England, Germany, Italy, Japan, Korea, Mexico, Spain, and Turkey. Awards that Randy has received include ADSA AFIA Dairy Nutrition Award, ADSA Pioneer Hybrid Forage Award, ADSA DeLaval Dairy Extension Award, ADSA Nutrition Professionals Applied Dairy Nutrition Award, and UW-Madison Cal's Pound Extension Excellent Award. Well done, Dr. Shaver. I will now turn the webinar over to Randy.
Thank you for involving me in the Nutritionist webinar. I'm Randy Shaver here at the University of Wisconsin-Madison, and I'm going to offer a perspective on starch digestion in dairy cattle. Been a lot of work in ruminants with starch digestion, primarily in beef cattle, many years ago, but this is still a relatively new focus in dairy cattle nutrition, and we'll try to cover some of the predominant focal points on that topic, with the emphasis being on lactating dairy cows. First, would like to talk about starch and where it fits into the picture. TMRs, total mixed rations for dairy cows, are approximately 70% carbohydrates. And a major portion of that would be neutral detergent fiber, but also a major portion of that would be starch. So if we think of dairy cattle rations being approximately 30% neutral detergent fiber or NDF, the starch could be anywhere between 20 and 30%, depending on the formulation of diets for forage and non-forage fiber sources, as well as grain sources. So you can see that a large portion of the total carbohydrates is being contributed through starch. To focus on starch digestion in ruminants, certainly the rumen or the first stomach has a major portion of the starch digestion. That is a microbial fermentation of the starch to volatile fatty acids. Certainly on higher starch diets, we would expect a greater proportion of the VFA or volatile fatty acids to be in the form of propionate, which is our main precursor of glucose through the liver and gluconeogenesis. But also the energy from starch does contribute to microbial protein synthesis. So starch does affect both energy and protein status of lactating dairy cows. We then have another shot at digestion of starch in the diet, and that being in the small intestine for any of the starch that escapes digestion in the rumen. And then any that would escape through the small intestine may, and the small intestine being enzymatic acid hydrolysis and enzymatic digestion to glucose for absorption. And then any starch that would escape both the rumen and the small intestine would have potential to be fermented in the hind gut. And so the digestion of starch to glucose in the small intestine would just contribute to energy. And also the microbial fermentation in the hind gut would supply VFAs, but would really have no impact on microbial protein or the protein status of the cow. Depending on the type of grains that we're feeding and also level of intake or rate of passage, we can see between 50 and 80% of the starch that's digested occurring in the rumen. And then certainly what's not digested there, we can have some post-ruminal compensation, so that starch digestibility would typically be 90% or greater in the total tract for lactating dairy cows, with the range being from approximately 80% to virtually 100%. So just one other thing I might like to mention is that the energy from starch that's digested in the rumen certainly contributes energy for the cow and also energy for microbial protein synthesis. The glucose that's absorbed in the small intestine really has not been shown to contribute greatly to improving production of either beef or dairy cattle. And it looks like some of that glucose would be used by the gut tissue, and also some of that energy that's absorbed in the small intestine may be directed into internal body fat. So it's one of the things that we need to keep in mind, that high ruminal digestion is a good thing for the cow until we get into very high starch diets or very rapid starch digestibility that may cause adverse effects on rumen pH, fiber digestibility, and possibly even slip into some forms of rumen acidosis. This is a slide from a meta-analysis performed by one of my students showing a relationship between ruminal starch digestibility and total tract starch digestibility. And we can see, as you might expect, if we have a very high ruminal digestibility, then we do see very high total tract starch digestibility. But also you can see with low ruminal digestibilities, some of that starch may still be digested. And so even though we have lower ruminal starch digestibility, total tract starch digestion can still be in the area of 80–85%. So just one of the things to keep in mind, that we do see post-ruminal starch digestion, but a pretty good relationship, certainly on the high end, between ruminal and total tract starch digestion. Why is it important? This is some results from the meta-analyses that agrees with some previous work done by Dr. Jeff Firkins at Ohio State University, showing a relationship between total tract starch digestibility and milk yield in dairy cows. And as you can see, as we go from approximately 80% total tract starch digestion up to 98%, virtually 100%, we're looking about a 2 to 2.5 kilogram response in milk production. And that's at very much the same NDF and the same dietary starch concentration. And so just an effect of improving total tract starch digestion for the cow. So a very important topic as we look at trying to optimize results that we get from carbohydrates in the ration. I'd like to talk a bit about forages, as that was an intended focus of the webinar. And just keep in mind that starch would not be coming from legumes or grass forage, but would be coming primarily from our starch-containing forages, or those which have grain in them. So certainly corn silage is a major source of starch worldwide. In some situations, certainly sorghum forage would contribute some starch, and then also some of our small grain silages may be a bit higher in starch as well, depending on the stage of maturity at harvest. So let's start out with really focusing on whole plant corn silage. To do that, I like to work with a schematic that was put together by our agronomist corn specialist at Wisconsin, and essentially recognizes the importance of the ratio of grain to stover and the concentrations of starch and NDF that we see in corn silage. And from a feed testing standpoint and a feed evaluation standpoint, certainly the percentage of starch and the percentage NDF are very important in determining the nutritive value of corn silage. And we see on average here in the Midwest, our corn silage would be approximately 30% starch and 42% NDF. As we look or dig a little bit deeper into the nutritive value of corn silage, we certainly want to look at the digestibility of both the yellow fraction or the kernel, and then also the green fraction or the stover. And the main emphasis of our talk today is on starch and the factors that affect the total tract starch digestibility in corn silage would be kernel particle size, so processing. The length of time in storage is very important in allowing for very high starch digestibility. The stage of maturity at harvest, and certainly length of time in storage and finer processing can override some of the negative consequences of late maturity harvest. The properties of the endosperm would be a bit more experimental here in North America, but certainly as we go around the world and into South America, we have this issue of flinty hybrids versus dent, or even flinty or dent versus flowery. The flowery being more of an experimental situation at this point in North America. And then some additives are being experimented with to try to improve starch digestibility in whole plant corn silage and also high moisture corn. I would like to mention briefly, just as we focus on forage quality, this trade-off between quality and quantity, or quality and yield. And this graph is certainly has application across forage type. We could look at a grass or a legume at a very early stage, at an optimal stage, and then at a very late maturity stage. And for corn silage, we would be looking at potentially an early milk stage, then a more optimum stage that may be half milk line or 3/4 milk line, and then finally a black layer stage or physiological maturity, which is closer to a high moisture corn stage of harvest in many situations. And you can see as we increase maturity on forages, we do see an increase in yield. This is dry matter yield in tons per acre. It could be in terms of metric tons per hectare, but this is in tons per acre in yield, and we can see that an increase in yield. But we can also see that what's increasing as we delay harvest is the yield of indigestible dry matter. So possibly hard kernels that are less digestible for starch, and then also stover, where the fiber becomes less digestible. And all forages have a sweet spot, so to speak, for harvesting the highest yield of digestible nutrients. And we can see that that shows up for corn silage typically at about a half to 2/3 or 3/4 milk line stage of maturity, where we get the highest total yields and the highest yield of digestible dry matter from that whole plant corn silage. And as we delay past that stage and closer to black layer or beyond, we do basically not harvest more yield or significantly more yield, and what we are harvesting here is less digestible nutrients and more indigestible material. So just want to emphasize that corn silage, like other forages, does have an optimal stage of harvest. So let's talk a bit about harvest practices and how that may influence the digestibility of starch in corn silage. And this is from a separate meta-analysis that my student had performed several years ago now, or a few years ago now, looking at an interaction that we found between kernel processing and stage of maturity. Stage of maturity here is captured by the whole plant dry matter content at the time of harvest, and the red bars would be the unprocessed material, and the more pink bars would be, or the lighter colored red, would be the processed silage. And we see that from 32% whole plant dry matter up to 40%, a nice response, a significant increase in starch digestibility due to processing of that kernel. And then as we go back to a wetter stage of maturity, that kernel is softer. It is more apt to be broken apart during normal chopping, and we don't really see a benefit of kernel processing. And then as we come up to a very dry harvest, still a numerical increase here for the processed silage to have a higher starch digestion, but it's certainly more variable, likely because of the volume of material that is being put through that roller mill, the setting on that processor, and we just have a tougher time getting good kernel processing. But this really shows that if we're going to use a processor, we can certainly get up into this 35% or higher starch whole plant dry matter and still have high starch digestibility. This also shows a relationship with the length of cut setting, or theoretical length of cut on the chopper, and the kernel processing. And we can see that the processing here goes from roughly a ½ centimeter theoretical length of cut up to well over an inch theoretical length of cut setting. And we'll see that the kernel processing did improve starch digestibility from about 1 centimeter up to 2.5 centimeters or slightly greater, as we went to much longer lengths of cut with traditional processors, or what we call conventional processors. No significant improvement here in starch digestibility by processing. And as we go back on the very fine chop, ½ centimeter theoretical length of cut, really no benefit of processing. It was chopped so fine that starch digestibility was not improved. So certainly length of cut is very important. Many of the processors would be close to 19 millimeter theoretical length of cut. There has been some interest with some processors of increasing that to 26 millimeter theoretical length of cut, or even to 30. and still trying to get good processing. There can also be differences in how that processor is set, and then there's certainly differences in whole plant dry matter. I just wanted to share with you a method of being able to evaluate, at least subjectively in the field, kernel processing, and this is available through a web posting at the Team 4-H website here at Wisconsin, put together by two of our ag engineers, Kevin Shinners and Brian Holmes. And it's basically a flotation procedure where the fresh chopped whole plant material is placed into a tub of water. It's agitated to allow that to separate with the fiber stover allowed to float, and the kernels, being more dense, will sink to the bottom of that tub of water. And you can then simply skim off the fibrous material and pour off the water, which allows you to take a look at the kernel fraction, which is what we are interested in from the standpoint of starch digestibility. And this graphic would show, again, what that material looks like. It does separate quite cleanly in a fresh material between the fibrous stover material and the grain fraction. It does not work, unfortunately, very well in an ensiled material coming out of the silo. So this is really a nice procedure to use when assessing the setting of the chopper, both for length of cut as well as the kernel processor setting. And then if we look at this material, we can see the material on the left is very coarse, not very well processed, a little bit better on this sample in the middle. And then lastly, the sample on the far right would really be the only one that we might consider being adequately processed, with all of those kernels having some degree of kernel damage and many of those kernels being fractionated. A less subjective procedure is a laboratory procedure that can be run, which is termed kernel processing score. And this involves sending a sample to a laboratory. They need to dry that sample so that the floury starch does not cling to the coarser fibrous material. And then that dried whole plant corn sample is dry sieved through a Rotap shaker and separated into very fine up to more coarse type material on those screens. And then a starch analysis is done on the starch, which is retained on a 4.75 millimeter screen, that being quite coarse, or the starch that passes through that screen, that being much finer. And kernel processing score is an index of greater than 70% passing through the 4.75 millimeter sieve would be categorized as excellent. Anything between 50 and 70% passing would be considered adequate for processing, and then samples that are less than 50% would be considered very poorly processed. This is a schematic that I put together. This is not a regression, but again, it simply looks at what could we expect if we improved a sample from poor to adequate or from adequate to excellent. And it's essentially 1 kilogram of milk that we could receive, or we could potentially remove a kilogram of grain, corn grain, as a starch source from the diet if we elected to go that route. And essentially, as we go from poor to adequate, it's worth a kilo of milk or a kilo of corn, and as we go from adequate to excellent, the same. So again, a very important effect of proper processing and chopping of whole plant corn. Unfortunately, we had done some survey work over the years through the feed testing labs, 3 separate laboratories over a number of years, up to 2012. And we can see that very few samples, in many cases less than 10%, were falling, certainly under 20% were falling into the excellent category. And unfortunately, in a number of cases, quite a number of samples were falling into the poor category. You can see 42% in two of these summaries. So we certainly felt from this that we could do a better job of chopper setup, chopper maintenance, monitoring the process, and try to improve from poor to adequate or adequate to excellent. The good news is laboratory, Ralph Ward was able to share from his laboratory some data, and I believe other labs have presented similar type data, where we have made progress from 2006 to 2014. We can see that about 2011, we do see a trend for improvements in the percentage of optimally processed samples, and a trend for a decrease in the percentage of poorly processed samples. And certainly the industry has made a real effort to try to improve the quality control of the harvest of corn silage. One of the reasons for this was some work that was done. We had done an initial study with a different type of processor. Just wanted to mention that. I think that's really what got the industry looking more closely at processing scores and trying to maintain a high degree of processing on corn silage. So I thought I would share this. This is done with a different processor than what's been conventionally used, or what was conventionally being used at that time. And we can certainly look at the results of our studies. We did two studies, one in 2011 and one in 2013. One with what we call a dual-purpose hybrid that could be harvested for silage or grain, and one with a low lignin or brown midrib mutant type hybrid. Our whole plant dry matter concentrations were 34% in the first study and 38% in the second study. We stored those in silo bags, and then we were allowing that to ferment in the first study for 1 month prior to starting our experiment, and for 4 months prior to starting our experiment in the second study. If we look at our length of cut settings, we'll see that our conventional material was 19 millimeters in trial 1 versus the shredlage at 30 millimeters in trial 2. 30 millimeters for the shredlage. As we come up to trial 2, the control was again 19 millimeter length of cut setting, and the shredlage was at 26 millimeters. We do see that the longer length of cut did result in an increase in the mean particle length. It resulted in a greater percentage of material on the top screen of the Penn State separator box. But if we look at the top two screens, which many nutritionists look at as an indicator of physically effective NDF, we didn't really see any improvements in that percentage of long material for the shredlage versus the control material. As we look at the setup of the rolls, we can see some differences in speed differential, with the shredlage being set in trial 1 at 30 versus 20%, so more of a aggressive tearing action of the rolls versus more of a crushing action. And in trial 2, the speed differential was virtually the same at 40% speed differential. If we look at processing score in the first study, 60 versus 75%, so certainly a difference in processing score. This, I should point out, for the control was still considered adequate processing. This would have fallen into the excellent category. And then as we come to trial 2, high adequate for the control versus an excellent processing score for the shredlage treatment. One thing of interest in the second study, even though we did not see a difference major difference in processing score, on average we did see a difference in the variance, a greater standard deviation for the control silage versus the shredlage treatment. As we go to the processing score, distribution, we can see that with the shredlage treatment in blue always being at high adequate or excellent, whereas the conventionally processed material had a number of samples that sat in the low adequate or mid adequate levels. So just a lot more variation in the conventionally processed versus the shredlage process treatment. If we look at the feeding trial, the days in milk would reflect mid-lactation at start, and if you take 3 to 4 month feeding period, essentially a mid-lactation experiment. The average milk production for the control cows was quite high, averaging 43 kilos or liters in trial 1 and 50 kilos or liters in trial 2. In terms of responsiveness, we saw no significant effects on dry matter intake. We did see significant increases in yield, 1 to 1.2 kilograms per day, and they were consistent across both studies, both in a dual purpose in trial 1 as well as a brown midrib low lignin hybrid in trial 2. Intake did increase slightly in relationship to milk, so unfortunately feed efficiency, milk per unit of intake was not affected. Milk composition, fat, and protein were unaffected, and we did see increases in component yields, which simply reflected the increase in actual milk yield. If we look at digestibility in the total tract for starch, and this would be in vivo data, so where we collected fecal samples from the cows on that experiment, we did see significant effects on total tract and ruminal digestibility, total tract being for the diet, ruminal being in situ measures of just the silage. In both experiments, as we go to NDF digestibility, we saw total tract NDF digestibility improved in the first study, but not the second. And we do see ruminal silage digestibility really not improved or not consistently improved in those two trials. Next, I'd like to focus on the second point on my schematic of corn silage nutritive value, and that is the effect of fermentation or ensiling, and also length of the fermentation period on starch digestibility. And generally, in our research trials, we certainly expect greater starch digestibility as length of time in storage increases. This is some data from one of our experiments. It's now published in the Professional Animal Scientist Journal of ARPAS, this data from an abstract. And we're looking here ammonia nitrogen concentration in the fresh material versus ensiled. So unensiled would be 0 months, and then months ensiled would be 1, 4, and 8. The blue being the ammonia nitrogen concentration, red being soluble crude protein concentration, and you see that increasing with ensiling, and it increasing as we go from 1 to 4 up to 8 months of ensiling. And then you can see that in vitro ruminal starch digestibility did increase as we go from unensiled to ensiled, and we do see that increasing as we go from 1 month in storage up to 8 months. And so this is certainly something very important that, like processing, can minimize some of the negative effects of delayed harvest on starch digestibility. This data just further showing that relationship between ammonia, nitrogen, and soluble crude protein. Reasonably good coefficients of variation or R-squared values as we go from low ammonia, nitrogen, and low soluble crude protein up to high. We do see that increase in ruminal in vitro starch digestibility. One of the things that we've noticed with kernel processing score is that it is also affected by length of time in storage. And this is likely because that starch protein matrix that we'll talk about is degraded during storage through effects of the protease enzymes that are produced or secreted by the microorganisms in the silage. We see that allowing not only an increase in starch digestibility, but also some breakage of the kernel. And so we can go from more of a vitreous kernel to a softer or more floury kernel, which does increase the processing score. And as you can see, in a vacuum sealed mini silo, as we go from unensiled to ensiled for 30 days, we do see an increase in processing score of about 4 percentage units. And then as we go from 30 days to 120, we now see that increasing further up to about 68% processing score, and then you'll see that leveling off. So I think it's very important that we look at processing score as we go through the feeding period to really determine what that silage is looking like from the standpoint of kernel particle size and potential starch digestibility. So some of that increase with time in storage is due to the breakage, likely breakage of that starch protein matrix due to fermentation, and some is due to a finer particle size as that starch or has that kernel is more open to breakage. I'd like to spend some time talking about the grain size side of the equation, and we'll use data from a meta-analysis that was performed by my student Luis Ferreto, a Brazilian student working on his PhD. And so a lot of the data that we showed was in a meta-analysis with whole plant corn silage literature publications, and this would be more specifically related to grain sources. First, looking at different grain sources, since not only corn would be fed as an energy source or grain source as we go around the country in the U.S. or around the world, but certainly barley or even wheat. And this data would show a lower ruminal starch digestibility for corn versus barley and wheat. And this is all data that's published in the literature with ruminally and intestinally cannulated dairy cows, where we can look at ruminal starch digestibility. So you can see much lower ruminal digestibility for corn, grain, and dairy cows than what we see with barley or wheat. As we go to total tract starch digestion, now we'll see that barley, corn, and wheat are much closer in total tract digestibility. And that's that aspect of starch digestion that we talked about at the outset of the webinar, where what starch that's not digested ruminally can be digested. Much of that can be digested post-ruminally. So our differences in total tract starch digestion and total energy available to the cow is much less for barley and corn and wheat than what we see in ruminal digestibility. This is largely due to differences in the starch fraction and the starch protein matrix, and we talk about prolamine proteins and the impact that they can have on digestibility of our grain sources. And we can see that these prolamines are very different as we go from barley and wheat or oats to corn or sorghum. We talk a lot about zein protein being kind of the aspect of starch digestion in corn that we're most interested in, but you can see very different prolamine proteins for the different grains. And we can also see that the level or concentration of those prolamines and their degradation is very different. Very low concentrations for barley, much higher concentrations for corn. And then as we go to something like milo, very high concentrations. And that really relates to some of the differences in ruminal digestibility that we discussed on the previous slide. And this slide put together by my colleague, Pat Hoffman, when he was still working in this area at the University of Wisconsin. We can look specifically at corn, and we see that prolamine or zein protein is our main aspect of starch digestion that we're interested in. But unfortunately, it's a very complicated area with really 4 types of zein protein, and it may not be the total concentration that influences digestibility of starch, but the different forms of that zein protein. But these prolamine proteins, can certainly cross-link or form a matrix with the starch that makes them less digestible. And we often talk about this prolamine protein being a lot like the lignin that we see in forages. Lignin tends to cross-link with cellulose and reducing fiber digestibility, much like these zein proteins or zein protein fractions can form this matrix with starch. And this does advance with maturity. So as we get to dry corn, it's much more important than in high moisture corn or corn silage. As we get to drier high moisture corn, it's going to be more important than in wetter high moisture corn. And then also as we get into corn silage, that typically is harvested at a much wetter stage of maturity than high moisture corn. But if we miss the harvest and get that too dry, we would expect some of these zein proteins to increase and really reduce starch digestibility potential in that forage. There are certainly some genetic differences. We would expect some of the flint corns to be very high in prolamine, and that helps to explain the lower starch digestibility for them. As we get into some of the normal dent corns that we see, really not a lot of variation, and these can be very moderate in prolamine protein. And then as we get into some of the floury opaque corns, they're very low in vitreousness, very low in prolamine proteins as well. So it's certainly an aspect of starch digestion that's going to be most important in our dry corn grains. So just a schematic, again, adapted from a slide put together by Pat Hoffman a number of years ago. We're focusing now on corn grain, and really the most important aspect would be processing. Particle size or fine grinding certainly improves starch digestibility, can override some of the adverse effects that we just discussed relative to endosperm properties. Another approach would be steam treatment or steam flaking, which does certainly have the ability to improve starch digestibility. As we go into harvest and storage, as I just mentioned, high moisture corn tends to be harvested at an earlier stage. It does go through fermentation, and much like the slides that I showed you for corn silage, length of time in storage does help to reduce that starch protein matrix. It does help to reduce the particle size, and we do see that improvement in starch digestibility with length of time in storage. And then, as we mentioned, certainly the endosperm properties is a part of the picture, but does tend to get masked or minimized due to effects of length of fermentation of high moisture corn and then degree of kernel processing. This is just a slide that was put together from our meta-analyses that we discussed earlier, looking at differences between high moisture corn, steam flake corn, and then dry rolled or dry ground shelled corn. And we'll see if we look at ruminal starch digestibility, the high moisture corn would be the highest, so certainly a faster rate and a greater extent of digestion in high moisture corn on average. Steam flake corn would be intermediate between high moisture corn and dry corn. If we look at total tract starch digestibility, the highest values numerically and statistically would be in favor of the high moisture corn and the steam flake corn. So certainly, again, some hindgut compensation for the steam flake, with less of that being digested ruminally and more post-ruminally. And then the dry corn has less total tract starch digestion in relationship to the lower ruminal starch digestion. Again, just to reemphasize this difference in the starch-protein matrix, just a slide to show the floury endosperm with greater voids between the starch granules to allow for greater enzymatic action, greater degradation of that endosperm in the rumens, so greater ruminal digestibility, versus the vitreous endosperm where you can see that starch-protein matrix really filling those voids and creating a situation with that endosperm or that starch endosperm being less digestible. One way around that we see is with steam flaking, as I mentioned. This is some data put together by Richard Zinn and Fred Owens. This data from dairy cattle, a lot of work done with beef cattle, but I'm presenting here the lactating dairy cow work. And you can see in general, as you reduce the density from 40 or 45 pounds per bushel back to 28 pounds per bushel, so a thinner flake, a lower flake density, we see much greater total tract starch digestibility. So the data that I showed you comparing dry corn to high moisture corn to steam flake was overall an average, but you can see steam flake corn, if we do a good job with the process, can have very high starch digestibility. If we do a poor job, can have very low starch digestibility with that very coarse flake. If we look at corn grain in a dry form, the major aspect that we can control in most situations is the particle size. And you can see as we go from a very coarse grind, 3,000 to 4,000 micron mean particle size back to 500, we see an increase here in total tract starch digestibility and hence total energy available to the cow. And we were able to do a regression analysis on that. You'll see the highest starch digestibility here at 500 microns, and then as you progress along that line, a reduction in total tract starch digestibility. So this particle size becomes very important, particularly for our flint corn, and then also for our dent corns that have a moderately high or higher than average vitreousness or prolamine concentration. This is a schematic that was developed from our feed grain evaluation system that is available on our website here at Wisconsin. And it basically looks at mean particle size of dry corn, again from 500 microns up to 2,000 microns. And we'll see a steady decline in total tract starch digestibility. And we'll see that very minimal effects of prolamine concentration or kernel endosperm type as we get back to that 500 microns or even 750 micron mean particle size, and it would only be with very coarse processing or grind size that we would see much of a spread as affected by the endosperm properties. We can look at that with dry corn. Again, a little more differences here in ruminal starch digestibility, but the major effect certainly is with kernel processing or mean particle size as we go from 500 down to 2,000. But with ruminal digestibility of dry corn, a little more spread related to the prolamine concentration or kernel vitreousness. And then we are able to look at a rate of digestion. The program, the spreadsheet will allow us to calculate that. And what we looked at was one prolamine concentration, very much average, very typical of 4%. And as we go from 400 micron mean particle size, the green, to 800 to 1200, we can see the rate of digestion, rate of starch digestion going from 22% per hour down to about 14%. So really, I think these slides would show something that we can manage very well and certainly minimize any of the adverse effects that we have of endosperm type is mean particle size. And we see that being the major effect here on total tract starch digestion, extent of ruminal digestion, and then also rate of digestion. As we go to high moisture corn, it is certainly affected by fermentation time. And this is some data that my student, again, put together for ruminal in vitro starch digestibility from laboratory samples. And we're able to see a relationship with the pH of that sample and also the dry matter percentage. So with wetter high moisture corn, less than 70% dry matter, and with a lower pH approaching 4, we see much higher ruminal in vitro starch digestibility. And that just really indicates that the extent of fermentation is very important in high moisture corn, the extent of silo fermentation. Again, much like with corn silage, now with a larger number of samples, we can relate ammonia nitrogen concentration here as a percent of crude protein and soluble crude protein as a percent of crude protein. And we'll see as to increase from low to high, we do see an increase and a fairly high R squareds on both of these, a very large steady increase here, linear increase in vitro ruminal starch digestibility as soluble crude protein and ammonia nitrogen increase. And those increase with a greater extent of fermentation in the silo. We can also apply the feed grain 2 evaluation system to high moisture corn, and we'll see again that at 1,000 microns, we really have minimal effects here of ammonia nitrogen concentration. As we get out to 1,500 or 2,000 microns mean particle size, we do see some spreads here, even in total tract starch digestion, from about 97% down to 94 to less than 95% in relationship to the ammonia nitrogen concentration as we go from very low, about 1%, so very short period of time in the silo, up to 7%, which would indicate either a wetter material or something that's certainly been in storage for a very long period of time. So ammonia nitrogen and mean particle size do have some major effects here in total tract starch digestion, and even more major effects as we look at ruminal starch digestibility, with a decrease here as we go from 1,500 to 2,000 microns, but then major effects as we go from 1% up to 5 or even 7% ammonia nitrogen concentration as a percent of crude protein. So really showing major effects on total tract starch digestion and on ruminal starch digestion, certainly of ammonia nitrogen or fermentation time, and then also a little less major, but certainly real effects of kernel processing. And then lastly, as we go to rate of digestion, again, we're going to use a more typical mean particle size for high moisture corn. 1,500 microns would be most common here in the Midwest. As we go from 2% ammonia nitrogen, so not a very great extent of fermentation, up to 6%, so a pretty major extent of silo fermentation. You see that rate increasing here from 18% up to 26%. So again, really showing major effects here when we try to model fermentation time as well as particle size in high moisture corn. The next area that I would like to talk about really shifts from some of these analytical approaches, such as ammonia nitrogen, rumen in vitro, starch digestion, soluble crude protein, dry matter, silage, pH, etc., and focuses more on what the cow can tell us by analyzing fecal starch. And we can really use that as a way to better manage starch digestibility at the farm level. These are some results from a field trial that we conducted a few years ago. This has been done by others in their states. I know Charlie Sniffen presented some data at the dairy science meetings, really showing that if we go into the field, a very large percentage of the fecal samples, if we go into a high group of cows and take a subsample of that group, maybe 10 cows, and take a composite sample, most of those farms, 60% or even more, will have 3% fecal starch or less. But we can see that 40% of the farms had more than 3% fecal starch, and a few farms, you can see percentage of samples, not very many farms, but we did see 11%, 16% on up to 20%. So there are opportunities to find these situations and then back up and look at the feed ingredients and see what might be done to improve starch digestibility. This would be samples from our research trials over the years that one of my students, Shane Ferdine, pulled together for a paper published in the Journal of Dairy Science. Again, you can see that 69% of those samples would have fell below 3% fecal starch, but we still saw 31% over 3% fecal starch, and we did see a number of samples sitting at 10% on up to greater than 14% of fecal starch. And this would be from experiments where we varied particle size of corn silage and grains, and we varied stage of maturity at harvest, and we varied diet composition. So we do have situations in lactating dairy cows where we can have fecal starch well above 3%. This is a regression equation that was developed from this data, and we were able to take our fecal starch data and our total track starch digestibility that was determined by looking at a indigestible marker in the feed as well as the fecal material versus the nutrients, and we were able to predict starch digestibility directly from fecal starch concentration. And you can see this equation: as we get less than 3% fecal starch on a dry matter basis, starch digestibility is approaching 100%. As we get to 5% fecal starch and less, we'll see starch digestibility is declining down to low of about 80%. And that checks very closely with what we've seen in that meta-analysis for a range in total track starch digestibility. So how might we use fecal sampling on farms? It can be used to directly predict total track starch digestibility, so we don't necessarily need to spend a lot of extra analysis time and money on rare earth markers or lignin or indigestible NDF to get at starch digestibility. We can use it to monitor specific groups over time, which is what I would recommend to, on a monthly basis, monitor the high group and to see when changes occur with dramatic reductions or increases in fecal starch, and try to relate that to changes in the feed that's in the silo or what's being fed at a given time. We need to remember that that reflects the total starch in the diet and not a specific feedstuff. So if we have both high moisture corn and corn silage and dry corn grain as our starch sources, those all will be contributing to those potential differences in starch digestibility over time. If we have less than 3% fecal starch, from the standpoint of starch digestibility optimum or maximums, we don't necessarily need to worry about the other ingredients. We certainly can look at those different ingredients, corn silage and corn grain, if we have very high fecal starch, and see if we can do something from the standpoint of better processing or maybe changing the proportions in the diet. From a low fecal starch, it really tells us that we probably can't do improved processing to improve starch digestibility, but it really doesn't tell us anything related to ruminal digestibility, rate of rumen digestion, and how that might be impacting milk fat percent and rumen acidosis. So on the low fecal starch, we still need to look a bit closer at the cows and make sure we don't have problems with starch digestibility being too great in the rumen. But on the high fecal starch, it does suggest a situation where we can certainly start to look at the feeds and maybe make some interventions in how those feeds are being processed or fed at the farm. Some work really provided, this slide provided by my former student Shane Ferdine. This was actually some Illinois work conducted through Rock River Laboratories a few years ago now, or last year, presented at the dairy science meetings, and basically shown if we want to use fecal samples to try to get at starch digestibility, we need to be keeping those samples cold as we transfer them from the farm to the lab. And you can see in this data, in the non-cooled samples, there was a 24% reduction in fecal starch versus those samples that were kept cold or on ice in the transfer from the field to the lab. You see that difference here, 5.4 versus 4.1%. So something to keep in mind as we try to use fecal samples to estimate starch and nutrient digestibility. Ruminal starch digestibility has been mentioned several times throughout my talk. At this point, it's really one of our only measures that we have to analyze specific feedstuffs or even a total mix ration for starch digestion. This can be done with batch in vitro procedures. Some will look at gas production. But the central ruminal in vitro batch procedure really dates back to a paper in the beef area published back in the mid-90s. And they looked at certainly aspects of starch digestion like particle size of the sample, the donor animal, etc., to try to optimize that procedure. And some variation of this is largely what's being utilized by the feed testing laboratories. It's really the only procedure that we have at this point to compare samples, but I just want to mention some of the shortcomings and why I think it's maybe more of an indexing of high, medium, and low. And again, like fecal starch, a way of looking at feedstuffs to determine whether we need to make some interventions rather than a direct measure of starch digestibility that we can use in ration balancing software. One of the challenges is the incubation length. The common time points have been 3 and 7 hours in vitro or 12 hours in situ. But those certainly ignore differences in rate of passage that can exist in your client herds, and also differences in rate of digestion that we might be able to get at if we had multiple time points. So this is one area where more work could be done to better utilize the in vitro procedure or gas production procedure to get at rates of digestion and maybe more realistic data from the standpoint of rate of passage or the KP in the herds that are being fed. Another challenge of these procedures is that they are typically ground through anywhere to a 2 to 6 millimeter grind, depending on the laboratory that we're utilizing. And that unfortunately can mask many of the effects that we discussed due to particle size on starch digestibility that certainly exist in your client herds. So the particle size differences for corn silage and high moisture corn and corn grain, much of that might be masked, and again, making these maybe more of a comparative index value over time or between samples rather than giving direct measures that we can use in ration software programs. Another big area that I think is open to more research would be the diet of the fluid donor cows, or the cow that's being used for in situ measures, and how that might impact starch digestibility ruminally, and how that might differ for the client herds that you're working with. Specifically, starch concentration and the source of starch has been shown in some research trials to affect amylase activity, which does affect starch digestibility. Again, making these values more relative versus more dynamic in our ability to use those. Certainly mentioned here, and then also NIR calibrations, while very robust and having much utility for our measures of starch, NDF, much less so when we get into in vitro starch or in vitro type measures. So how might we utilize these numbers? I certainly mentioned from an indexing standpoint, high, medium, and low, determining when they change and whether we can make some interventions. Certainly research and application of these values to incorporate into our models for adjusting our energy values. And so this is an area where we've used something like a processing adjustment factor. Might it be possible to use rumen digestibility to predict changes in total tract starch digestion and allow that to affect our TDN or TDN1X or NEL values? I think this is more an exploratory experimental stage at this point. And then finally, maybe making some adjustments on rumen digestible starch. And I do show an example here of 15 pounds of corn A, 70% starch. This is 15 pounds of dry matter, a 60% in vitro ruminal starch digestibility. If we multiply those out, would come to 6.3 pounds of rumen available starch. And then if we go back to a different corn, the same starch concentration, but a higher ruminal in vitro starch digestibility, 75 versus 70, then we would need to feed only 12 pounds of dry matter to get the same intake of rumen available starch of 6.3 pounds. So this is just one area that we might be able to incorporate some of these values within a herd or within a feeding situation to do a better job with our feeding rates on corn, for example, or even corn silage as we move down the road. So with that, I would like to close the webinar. Thanks for participating, and point out to stay tuned. This area of starch digestibility is an active area of research and field application, and I'm sure that we will learn a lot more as we go forward. Thank you.
Thank you, Dr. Shaver, for that terrific and timely presentation. I want to remind our listeners to attend the next webinar to be held on the 12th of August at 6 p.m. UTC-4. Our presenter will be Dr. Rick Grant. He will speak on research that has been performed at the Minor Institute as well as applying research at the farm level. Rick Grant has served as the president of the Minor Institute since February 2003. Prior to that time, Rick was a professor of ruminant nutrition and extension dairy specialist at the University of Nebraska-Lincoln. His primary areas of interest include forage and non-forage sources of fiber, carbohydrate nutrition, and dairy cattle behavior. He received his BS in animal science from Cornell University and his PhD in ruminant nutrition from Purdue University. Rick was also a postdoctoral research associate at the U.S. Dairy Forage Research Center in the University of Wisconsin prior to becoming a professor at the University of Nebraska. Rick's primary role is to provide leadership to institute staff as they work towards a common goal of optimizing the crop-animal interface through research and educational programs. Save the date and time, Wednesday, August 12th at 6 p.m. Eastern Daylight Time, or UTC-4. I would like to thank the people who made this possible: Tom Tolucci at AMTS, USA and Global, Marcelo Hens Ramos at 3R Labs Brazil, Paula Torillo in Argentina, and our translators at each location. We also wish to thank the sponsors of our English language seminar, Ajinomoto Heartland Incorporators, maker of AgiPro L rumen-protected lysine, and Virtus Nutrition, manufacturers of Energy 2, Prequel, and Strata. Archived presentations are available at our website. Ordinarily, we have a question and answer period at this time. Due to some recording issues, we are unable to archive the session from this webinar. We lost the entire recording, but Dr. Shaver was gracious enough to re-record his presentation for the website archive. Thank you for listening, and good night.